Literally, the options are all right. So this is a follow-up to Sarim's Genetics 101 course. I'm introducing CRISPR to you guys, and CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. It's a new gene editing tool that's come about in the last decade, and in the synthetic biology course, we also went over whether it's a biotechnology tool or whether it's a synthetic biology tool. It's been argued both ways. CRISPR is a naturally occurring system in bacteria and was considered a biotechnology tool and when it was used in that way. But since the Dorner lab has intervened, used it in eukaryotic cells, it has been considered a synthetic biology tool and has been used in that capacity since then. The central dogma of, for some context from Sarim's presentation itself, the central dogma of life is when DNA is transcribed into mRNA, and which is in turn translated into proteins. So this means that DNA is a double-stranded code of life, and the process by which the DNA is read and transcribed into proteins is a two-step process. So DNA has adenine, thymine, guanine, and cytosine, four nucleotide bases that make up the basis of all life on Earth. mRNA has adenine, guanine, as well as cytosine, but instead of thymine, they have uracil, and that is how it is translated. It is read from the 5' to the 3' end and translated with their complementary bases in the next step. And in order to read the mRNA, you need the table on the right. So when you're looking up, which combination, which 3-letter combination you're looking in the codon, the start codons will indicate where the genes you can start reading the gene, and the stop codon will tell you when to stop reading the gene, and when the protein will be transcribed at that point. And the last is the translation, when the mRNA is translated into the protein itself, into the amino acid protein. And like I said before, it is the 3-letter combinations that make up these polypeptides. Now, the repair mechanisms. So all human beings, almost all eukaryotes on Earth, have a DNA repair mechanism that is inherent to the being itself. It is usually when there is a single-stranded break, there are a few mechanisms that just kick in instantaneously and are a lot less energy expensive as compared to double-stranded breaks. A double-stranded break is the worst kind of damage that can be done in a cell, especially in a nucleus, and they have two mechanisms to tackle it. One is called the non-homologous end joining, which means that when there is a double-stranded break that occurs, like in the left-hand side picture, it just basically ligates itself together. This is the most commonly used double-stranded mechanism, but it is also the most error-prone. In CRISPR, what we exploit is something called the homologous directed repair, which means that when there are two chromosomes, one from each parent, the error that needs to be corrected is looked into the sister chromosome and copied into the new pair. Right. So over here, you can see that this bright purple is what needs to be repaired into the double-stranded break. It has been copied from the sister chromatid, and these dark purple are actually the sequences which are adjacent and are similar to the repaired sequence. Does that make sense?
Yes, it does.
Okay. So when this is attached, it makes a perfectly corrected copy of the gene. There is a TED video over here which will explain the same thing. For single-stranded mechanisms, I've put together a resource sheet that I will link at the end of the presentation.
In just one of your cells gets damaged tens of thousands of times per day. Multiply that by your body's 100 trillion or so cells, and you've got a quintillion DNA errors every day. And because DNA provides the blueprint for the proteins your cells need to function, damage causes serious problems, such as cancer. The errors come in different forms. Sometimes nucleotides, DNA's building blocks, get damaged. Other times nucleotides get matched up incorrectly, causing mutations. And nicks in one or both strands can interfere with DNA replication, or even cause sections of DNA to get mixed up. Fortunately, your cells have ways of fixing most of these problems, most of the time. These repair pathways all rely on specialized enzymes. Different ones respond to different types of damage. One common error is base mismatches. Each nucleotide contains a base, and during DNA replication, the enzyme DNA polymerase is supposed to bring in the right partner to pair with every base on each template strand, adenine with thymine and guanine with cytosine. But about once every 100,000 additions, it makes a mistake. The enzyme catches most of these right away and cuts off a few nucleotides and replaces them with the correct ones. And just in case it missed a few, a second set of proteins comes behind it to check. If they find a mismatch, they cut out the incorrect nucleotide and replace it. This is called mismatch repair. Together, these two systems reduce the number of base mismatch errors to about 1 in 1 billion. But DNA can get damaged after replication too. Lots of different molecules can cause chemical changes to nucleotides. Some of these come from environmental exposure, like certain compounds in tobacco smoke. But others are molecules that are found in cells naturally, like hydrogen peroxide. Certain chemical changes are so common that they have specific enzymes assigned to reverse the damage. But the cell also has more general repair pathways. If just one base is damaged, it can usually be fixed by a process called base excision.
So this is the base excision repair is usually what happens with a single-stranded, break. The double-stranded is these two major pathways.
Excision repair. One enzyme snips out the damaged base, and other enzymes come in to trim around the site and replace the nucleotides. UV light can cause damage that's a little harder to fix. Sometimes it causes two adjacent nucleotides to stick together, distorting the DNA's double helix shape. Damage like this requires a more complex process called nucleotide excision repair. A team of proteins removes a long strand of 24 or so nucleotides and replaces them with fresh ones. Very high-frequency radiation, like gamma rays and X-rays, cause a different kind of damage. They can actually sever one or both strands of the DNA backbone. Double-strand breaks are the most dangerous. Even one can cause cell death. The two most common pathways for repairing double-strand breaks are called homologous recombination and non-homologous end joining. Homologous recombination uses an undamaged section of similar DNA as a template. Enzymes interlace the damaged and undamaged strands, get them to exchange sequences of nucleotides, and finally fill in the missing gaps to end up with two complete double-stranded segments. Non-homologous end joining, on the other hand, doesn't rely on a template. Instead, a series of proteins trims off a few nucleotides and then fuses the broken ends back together. This process isn't as accurate. It can cause genes to get mixed up or moved around, but it's useful when sister DNA isn't available. Of course, changes to DNA aren't always bad. Beneficial mutations can allow a species to evolve, but most of the time, we want DNA to stay the same. Defects in DNA repair are associated with premature aging and many kinds of cancer. So if you're looking for a fountain of youth, it's already operating in your cells, billions and billions of times a day.
Hey, yes. So the interlacing of the two strands between the sister chromatid and the actual damaged DNA is what is called homology-directed repair. Again, what we are exploiting in CRISPR. It simply reads and complements the sequences that it sees in the sister chromatid in order to make a perfect template and repair the double-stranded break. And why this is important is because it is what we will be using in order to design our own sgRNAs, so single guide RNAs. So malaria is a disease where the Plasmodium parasite uses mosquitoes as a host and propagates by using human red blood cells as an incubator. The reason I'm bringing this up is because there have been both a CRISPR gene drive that has been made for malaria, as well as a CRISPR cure for sickle cell anemia. And sickle cell anemia was actually nature's evolutionary response to malaria. So malaria was endemic in sub-Saharan Africa. Eradication efforts were fierce, with mosquito sprays, nets, and other protective gear being shipped over. Almost 3,000 to 7,000 children died every year from the disease from around early 2000s through 2010. Now, sickle cell anemia was the evolutionary mechanism to malaria. It evolved in human beings as a defense mechanism to malaria, which uses our red blood cells. So by taking on the sickle cell shape, it kind of inhibited the parasite in using the RBCs. The problem arises only when there were two recessive copies of the sickle cell gene. So if the HBB gene is what determine the shape of RBCs. So if both of these copies from both the mother and the father were recessive, it turned out that all the RBCs took on the sickle cell shape, and they clogged the arteries by sticking together. But if only one, which means from a heterozygous pair, if only one was dominant, it had the proper RBC shape, whereas the other took on the sickle cell shape, it gave enough resistance to malaria for the organism or for the human being to survive. And in fact, it conferred some kind of immunity to the person who was infected with malaria. So the HPV gene was present just in one allele. So this is still observed in this protective element of sickle cell anemia is still observed in Africa and East Asia, as the number of malarial infections are still extremely high. So now the disease sickle cell anemia, again, when there is a homozygous property involved, a homozygous recessive element that is involved, which means that both of them have the sickle cell trait from both the mother as well as the father. So the problem arises when both of them carry the trait in their chromosomes, and it causes a malignant condition, which means that after releasing oxygen to the tissues, the mutated proteins, they lock together, they form a rigid row of individual proteins, and they deform the cell into like a long sickle shape. Blood cannot flow smoothly through the blood vessels, especially the arteries, and the lack of oxygen causes sharp stabbing pains across the body. These cells have a very short lifespan for about 20 days, and usually healthy cells, they survive for about 4 months. And the patients usually don't survive past 20 years of age if completely left untreated. So the long-term effects of sickle cell anemia are retinal detachment, stroke, difficulty breathing, and lowered immunity. The available therapy right now is hydroxyurea and bone marrow transplants. And both hydroxyurea is a chemical solution, whereas bone marrow transplants are usually from a close relative, usually a sibling, and it has a very high rate of rejection. So how are we using CRISPR in both malaria as well as sickle cell anemia? So to understand CRISPR, we need to understand the bacterial arms race. In the bacterial arms race, bacteria used to predate on human beings. Bacteria, like in nature's usual flow, bacteria has its usual predators, which are called bacteriophages, which are basically viruses that attack bacteria. Now, the evolutionary mechanism in bacteria which fight against these viruses, their genetic code, is called CRISPR. So the bacterial defense system is, they basically just cause one double-stranded break in the virus DNA, and that basically renders the virus DNA incompetent to to infect them for the next generation. Human beings have re-engineered both anti-CRISPR as well as CRISPR. Now, anti-CRISPR is basically what, again, another evolutionary mechanism within bacteriophages, which has evolved in order to fight, CRISPR in bacteria. These are just like some popular memes I put together. John Oliver has a great episode on CRISPR, which you can find online. So CRISPR sequences in the genomes of some prokaryotes. Some prokaryotes are usually single-celled organisms, mostly bacteria, that act as genomic record of a previous viral attack. So CRISPR, the beauty of the system is that even after a bacteriophage attack has taken place, it not just creates that double-stranded break and renders the virus, the bacteriophage DNA, completely incompetent. It also keeps a record of the virus attack in order for future attacks to be mitigated. So in its own sense, it is a bacterial defense system. So in order for CRISPR to work, it needs two things. It needs a gRNA, which is a guide RNA, as well as a CRISPR-associated protein or a Cas protein. Bacteria use the sequences to recognize and disarm the future invading viruses, which means the Cas needs to find its way. It's like a bloodhound which needs to find its way through the huge genetic sequence that we have, latch on to the correct point so the cut or the shredding can happen at the right point. And CRISPR-associated protein 9, Cas9, is the most important. It is the most prevalent, Cas, which is in our toolkit right now. It acts as a scissor. We have other Cas proteins as well, which act as shredders. But Cas9 is a scissor. It makes very precise edits in the place that we want. If you're able to engineer it to a point where we can take it to the right spot, Cas9 is very precise in what it does. So what Cas9 is a PAM sequence. A PAM sequence is a protospacer adjacent motif. So a protospacer is the area where a cleavage or a cut needs to occur within the double-stranded genetic code. Whereas the protospacer adjacent motif means that the motif that is adjacent to the protospacer, which simply means it is the area where the Cas or the bloodhound needs to take a hold of in order to make the cut right near. So if this is a genetic code, which I can show you here, a PAM sequence would be a repeated sequence where the Cas holds onto, and right next to it, adjacent to it, a cut will occur over here. So this is the CRRNA, and this is the tracer RNA. The tracer RNA is basically the backbone of the entire system, whereas the CRRNA is the CRISPR RNA, which will be like a 20 nucleotide sequence, which helps the PAM sequence bind to, which helps the Cas9 bind the PAM sequence. The template will basically be a complement, which will help it to find the place where it needs to make that cut. So in the natural system, in the natural CRISPR system, there are two basic phases. Phase one would be the immunization and acquisition of the bacteriophage sequence. So this means that the first time the bacteriophage attack occurs, it capturing that genetic sequence in order for it to remember is phase one, and which is immunization. And phase two is defense against the bacteriophage by cutting the genetic material. It captures the genetic code and then makes a cut, killing the bacteriophage. So protospacer is a DNA region targeted for cleavage by the CRISPR system. It is the point which is to be cut. Now, protospacer adjacent motif is a PAM sequence. It is also very important. It is the sequence motif, which is immediately adjacent to the protospacer sequence. And so now we know that almost every bacterial strain has a CRISPR system, whether it is Streptococcus pyogenes or Streptococcus like aureus, or any bacterial strain that you find, they will have their own immune system. They will have CRISPR in it. So every bacterial CRISPR system has its own PAM. So the most commonly used is the Cas9 sequence in Streptococcus pyogenes, which is NGG. So I have also taken the liberty of writing down a few more PAM sequences over here from different bacteria, which you guys can go over later. But here, N is any nucleotide, G, T, A, or C. And I will go over an exercise later. So now what is the guide RNA and what is a natural CRISPR system? So this picture on the left is the natural CRISPR system. This is a 20 nucleotide sequence, which is a CRISPR RNA. And it is usually the gRNA target sequence, which means it is the region to be cut by the CRISPR system. Tracer RNA is the backbone to the entire CRISPR system. And until now, we had not re-engineered the system. Synthetic biologists had not re-engineered the system until 2016 in the Doudna lab. So now when we re-engineer the system, we use HDR. HDR stood for homology directed repair, and it was customized by using two homologous ends and a sequence of our choice, the sequence being the template. So instead of the duplex gRNA sequence, a single gRNA sequence was synthesized. So how did they synthesize just one sequence instead of this 20 cRNA and tracer sequence? They just linked it together with this loop. The elegance of the system is the single guide RNA, sgRNA, or single guide RNA. Now, tracer RNA is a scaffolding, and crRNA is the customizable nucleotides like downstream. It is essentially the template. So the system was re-engineered by linking together the CRISPR RNA and the tracer RNA with this linker loop, and into a format that would provide the target information on one end of the transcript. So the target information is on the crRNA, and the structural handle for binding to the Cas9 on the other end. So over here, the Cas9 is bound, tracer RNA is over here. So it is essentially the backbone. So now to design your own sgRNA, what you need is a PAM sequence. If it is on the antisense strand, which means it is on the 3 to the 5 end, you need to start looking for the NGG sequence from the 5 to the 3. From here, you need to count 20 nucleotides towards the 5' end. And if you, if it is like a larger sequence, you can use other tools such as ChopChop, and I believe Benchling has a new tool which will help you design your own sgRNA. So now is the exercise. So PAM again is a protospacer adjacent motif. So if you need to find a protospacer adjacent motif, you need to find the protospacer first. So this is the area that we are targeting to be cleaved, and we need to find another PAM sequence. And once you find the PAM sequence, if you count 20 nucleotides towards the 5' end, you will find the CRISPR RNA as well. So these two are the same sequences. This is the other PAM sequence. And depending on, where you're looking, you need to count 20 nucleotides towards the 5' end, and that is a CRISPR RNA. Now the CRISPR toolbox, like I mentioned earlier, Cas9 is not the only protein that we're using in the CRISPR toolkit for re-engineering, bacterial systems. We're also using Cas12a, which is basically a detector diagnostic test. It targets DNA and reliably detects like the nucleic acids, especially if you're looking for SNPs. SNPs are single nucleotide polymorphisms, which means that in a long sequence of of a genetic code, we're not looking for a particular area where the genetic code is messed up. We're looking for one nucleotide which has been mismatched, and that is extremely hard. So we use Cas12a for the detector diagnostic test. The next one is Cas3. Now, Cas3 is a shredder. There is a startup on NC State's campus called Locus Biosciences who's using Cas3 in order to fight antibiotic resistance. Cas3 basically shreds the genetic code, and it renders the organism completely, incompetent, as well as in most situations dead. Now, Cas13 is used as a Sherlock diagnostic test. They did make a COVID-19 diagnostic test with CRISPR-Cas13 at the height of the pandemic. It targets the RNA instead of the DNA, and it reliably detects the nucleic acids of the virus itself. So now I would like to get back to the CRISPR therapy of malaria. Now, malaria we had to look into like fighting malaria genetically by either making a gene drive. So there were two approaches being taken to the gene drive. The Imperial College of London decided that they will start mutating a gene called double sex. Double sex is found in the female Anopheles gambiae mosquito, which and by like knocking out this particular gene, it makes for the development of the reproductive organs of the female mosquito. They don't develop their reproductive organs basically once you cut this gene. They cannot lay any more eggs, and their mouth is deformed, so they can't even eat. So they don't survive most of the time. They cannot draw blood from human beings, so they can't even if they do carry the parasite, they definitely can't spread it. So this combined with the gene drive, double sex is known as a homing endonuclease, which is a selfish gene. A homing endonuclease or a selfish gene make sure that this trait will definitely be passed on to the next few generations. And most of the offsprings will share the trait of the double sex gene being disabled, and the fact that they cannot develop their reproductive organs if and when they're able to proliferate. So we are essentially wiping out this strain of mosquitoes in sub-Saharan Africa. The ethical debate over here is how much are we allowed to play with nature, how much of the ecological balance can we actually disturbed before it comes back to bite us. So the question here being, can you justify wiping out an entire species for the betterment of humankind? Malaria here has been a prevalent problem. They've been looking for solutions since the early 2000s. So the other approach, which is considered the more humane, the more ethical approach, is by the University of California, Berkeley. Researchers here are creating mosquito vectors. They're disabling genes where they cannot transmit the plasmodium to human beings. So plasmodium in mosquitoes itself, they're not harmful to the mosquito, and by transmitting, they cannot transmit it to human beings. This is a gene drive that they are currently experimenting. It's still in the lab. It is a lot more complex to actually, look through this gene drive being transmitted and whether it will work. But the Imperial College of London, this is an approach which has been time tested, but is possibly not ethical. So this is definitely a discussion they're having in the genetic editing circles, whether we should wipe out an entire species for our own benefit. Now, sickle cell anemia, the CRISPR therapy, like I mentioned before, this is a disease which is caused by the replacement of one nucleotide, one base pair, not even one base pair, just like one nucleotide. The T, which I have which I've highlighted in red, this isn't supposed to be here. So it should be an A, it's supposed to be adenine. So CRISPR-based gene editing has been done on a person already. Her name was Victoria Gray, and in the United States, and it was treated with CRISPR. A single base pair mutation was in the HPV gene was replaced with the proper adenine. That the edits were reintroduced into the patient somatically, which means that it was made only for her and not her offsprings. And the chance of rejection by the body of this patient itself was extremely low. If a bone marrow transplant had been done, the chances of rejection were a lot higher. So the third approach over here would be to use CRISPR to turn on ferroglobin. It's a form of hemoglobin that can override the sickle cell gene altogether. That's research that is currently happening as well. So I've linked this particular article if you guys are interested. So now I would like to go into human germline editing versus somatic cell editing. So in Victoria Gray's case, we looked into somatic gene editing, which means gene editing within the patient without harming their germline or without changing the germline, the sex cells, in order to pass on that trait from one generation to the next. Instead, we are curbing the edits that we make to this one person, this one generation, possibly to help them live a healthier and better quality of life. But this example is Jian Kui. He is basically he was the first person in China who managed to make a germline edit. He used CRISPR and he managed to take out the receptor for HIV in a pair of twins. So these children are possibly less susceptible to HIV than the rest of us. And this trait that they have received from this edit, the CRISPR removal of HIV receptors, will be passed on to future generations, to their kids as well. It will not just be curbed to them. We do not know the evolutionary implications of this. And as far as papers have come out on these children, we know that there have been, their genetic code has large patches of mosaicism, which means large areas of genetic code which have been which are uneven or unruly between both of these children. Again, it does not seem to have any immediate effects because these children do seem to be healthy, but there were no clinical trials that were done before this, and they were children and whose lives were they were private citizens and they have private lives. Their father did have HIV, and he did consent to these children having the procedure, but they were too young to give consent themselves. So largely his scientific license was taken away, as well as he was jailed for 3 years. The technology is in its infancy, so how much we can experiment with it, especially on human beings, is very limited. How do evolutions turn malignant? We really don't know. It's like the sickle cell and malaria examples. Sickle cell was supposed to be a good evolutionary strategy by nature against malaria for people who have been rampaged by it. But now it has turned into a homozygous recessive trait that could kill people in a much lesser time as compared to malaria. So, knowing what we know about malaria as well as sickle cell anemia, how much do you think we should um, interfere with nature, and how much of germline or embryo editing should ever be legalized? So Jennifer Doudna has had like a evolving perspective on it. When this technology first came out, she had written many books taking like a hardcore stance against germline gene editing. The fact that we don't know how much of a trait will be passed on to children, uh, and how much we are supposed to interfere with nature was like a big concern at that point. But I believe she has slowly taken a stance away from that, and she's embracing the idea of germline gene editing, at least for therapy in like conditions such as Down syndrome, and things like that.
Neha,
Now,
quick question.
yeah,
Can you go back
yeah.
to the slide where you're talking about yeah, can this one actually. So you're talking about how nature, quote unquote, developed its own response to malaria, and that led to sickle cell anemia, right?
Right.
Why? My question here is, why doesn't or has nature not been provided enough, quote unquote, space to generate its own response to sickle cell anemia? Like, why didn't that happen?
Well, nature is a lot slower than us in the lab. To be honest, it takes like thousands of years for one particular mutation to turn up, and sometimes it's a game of chance. To be completely honest, um, sometimes mutation is good, sometimes mutation is bad. This we know is a response to malaria, because in people who had just one trait, like one allele which had the sickle cell HPV gene, they had like 50 to 70% chance lower chances of actually getting malaria as in comparison to the others. And this was like sickle cell anemia was more prevalent in the areas where malaria was like rampant at that point of time. So we know it was an evolutionary response, but no, nature has not had enough space to make its own defense mechanism against sickle cell anemia. That's the question.
Yeah, thank you for that.
Right. So this is Joshua Zena. He is a biohacker. He was a NASA scientist. He has a PhD from the University of Chicago. He turned a biohacker on his mission to democratize the access to CRISPR technologies and gene editing in general. He is actually here in Austin, and he has his own company is called the Odin. They're conducting experiments. His latest experiment is actually him changing his gender genetically into like a woman. Um, he's a very eccentric character, to be very honest. He has no consent, so he does experiments on himself, and he is also conducting experiments on like small animals like dogs, and he's tried to like switch off the MSTN gene in order to make himself more muscular. This is exactly what most ethical like books discuss. The fact that we are taking gene editing and we're making our own I don't know, our own engineered Gattaca. It's a movie if you guys haven't watched it. Um, while his intentions seem noble, I personally don't think it's a great way to go. To like satisfy our vanity before our necessities at first, especially if we do have such a powerful technology, it needs to be taken with a grain of salt. His argument is that the time to use and experiment with CRISPR is now. What his defining point is that even if you do have CRISPR therapies, which we do have right now for sickle cell anemia as well as for a spinal disorder, it will cost billions of dollars and will be only accessible to the richest of the population, which is how it is right now. Like insurance doesn't cover it, and only the top 10% can even afford the technology. Here, if we do not democratize it now, it will, natural selection will be taken over by the privilege of society. While he does have his points, his experiments right now are leaning more towards, vain experiments rather than therapeutic or like diagnostic experiments, which are more like necessary at this point. So now the patent battle. The patent battle is between the University of California, Berkeley, where Jennifer Doudna and her team first found CRISPR, and the Broad Institute of MIT and Harvard. They've been fighting for the legal rights to use CRISPR as well as get patent, money out of it. It's been It's between Zhang and Jennifer Doudna, and it's been since early August, like 2012, when Berkeley decided it has to like put in a little more money and push the patent forward in the process. Their patent forward in the process. In January 2016, the patent interference proceeding started, and in the same year, Doudna won the Doudna and Charpentier, they won the Nobel Prize in chemistry for CRISPR itself. It's recently it came out that Zhang won in the patent for CRISPR, and he also has a bunch of base excision therapies that are patented under his name. The Broad Institute now gets almost half the forwards from the use of CRISPR anywhere. The Innovative Genomics Institute, which is the Berkeley Institute, which where CRISPR was found, is still pursuing the patent, and this is like a hot topic because CRISPR is just one tool. We see a entire synthetic biology future where we can re-engineer it into different tools, into different organisms, and those patents will be another legal battle altogether. So the Coursera has this one course where they teach you how this needs to be addressed and how to like file your own patent for synthetic biology products, especially made with CRISPR. I'm linking that in the chat. So now CRISPR startups by application areas, again, as a testament to how important these patents are and how important these patent battles are the number of startups that have come out of the CRISPR technology phase as well as place. So there's biomedical R&D, Mammoth Biosciences. I think right there you pointed out that there was, a company that was using CRISPR to like bring the mammoth back, the woolly mammoth back. Again, they don't have enough of the genetic code to actually do that. What they're doing is taking an elephant genome and they're trying to insert what little they found from like excavations and see, what organism comes out of it. Again, there are a lot of ethical implications there. They have not gotten away scot-free. There have been a lot of discussions, a lot of investors contemplating whether this is the first and the most important use of CRISPR. It is being funded by George Church, who is widely considered the father of genetics or gene editing at least. On NC State's campus itself, we have two CRISPR startups, Locus Biosciences that is using CRISPR-Cas3 to fight antibiotic resistance, and TreeCo, which is using CRISPR in order to get more pulp out of every tree, so there's less deforestation in the world. They're doing these in greenhouses. Again, Syntego is a huge CRISPR company which has started making, just started making like sgRNAs that are tailor-made for particular experiments, particular researchers, and there is a lot of industrial biotech R&D which is happening in the space right now. Sherlock Biosciences is making like diagnostics in using CRISPR. They were the ones who made detector as well as the COVID-19 diagnostic test using CRISPR-Cas12a and CRISPR-Cas13 in this slide, this one. So 12a, 13, both of these are Sherlock devices. So yeah, agriculture is also another space where we have seen a lot of CRISPR implications. And considering the range of the technology, patents will be very important. And startups in synthetic biology are here to stay. Do you guys have any questions?
Can you go back to the last slide?
Okay.
So we have a lot of like genetically modified crops, right? The cotton, the brinjal, the eggplant, and all of that. So
That's interesting.
Yes. So how will gene editing like can you give me an example of like I understand editing the tree genome so as to get more pulp, that's something which will be interesting to reduce the amount of deforestation that we do. But is there something which should like for example like increase the content of vitamins or like ensures that we get like all the necessary nutrients, like let's say from one food, so that we don't need to, is there something like of that sort possible?
It is. So gene editing isn't a new concept. They've been doing this for like decades together right now. What we have been doing is use like ancient technologies like ZFNs, that zinc zinc finger nucleases. So they aren't more expensive per se, but they are harder to use. They're harder to program. So keeping all that in mind, agricultural gene editing has been kept to a minimum, especially because of the outcry with GMOs. But it's not publicly accepted GMOs. But with the advent of CRISPR, there's minimal intervention, and we kind of do get the products like you suggested. We do get, better vitamins, better. The nutrient content can be changed with by genetically manipulating the crops. Also, their weed resistance for like the farmers, or better fertilization. I mean, better like output from the same amount of seeds, things like that.
Yeah, like for
Mostly like,
example, like pest resistant stuff, right? Like for
yes,
example,
yeah.
locust infestations. We've all heard of how locust infestations can basically kill entire like scores and legions of crops, like across acres of farmland. And if you can have something which is like resistant to that or resistant to something else, and yeah, that's something that will be really interesting to see the future.
Any other questions?
Oh, that's it.
All right, thank you so much. And see you guys next Saturday.